This method can help answer key questions related to the biological control of soil-borne plant pathogens. The gold standard for testing materials like compost is to have a plant bioassay to test whether it's toxic to the plants. However, that takes 3 weeks. This method can be done in about 2 to 3 days. The main advantage of this technique is that it is simple, affordable, and effective.
This assay is a rapid screening of specific compost to suppress the growth of Rhizoctonia solani. However, this assay can also be used against other soil-borne plant pathogens, such as fusariums, pythiums, and phytophthora.
Visual demonstration of this method is critical because it takes care to isolate and maintain a pure culture of a fungus while avoiding contamination. Demonstrating the isolation procedure will be Taylor Reddyhaw, a graduate student from my laboratory. Emma Wright, an undergraduate student from my laboratory, will demonstrate the competition assay.
To begin, sow red radish seeds in soil known to have a history of disease caused by R. solani. After 3 to 4 weeks, remove the seedlings from the soil and rinse them with tap water. Using a razor blade, cut 1 centimeter segments of the hypocotyl and root that have a brown color. Then, use flame-sterilized forceps to dip the segments into 10% bleach solution for 1 minute. After this, rinse the segments in sterile water. Next,